hear us? Good, thumbs up. So thank you for the introduction, Lars. And again, thank you very much to SFI Visual Intelligence for helping us organize this event. So now let me introduce you to the first point of the program and our first speaker. His name is Rahmi Lale, and he will be talking about how it is to take the leap from being a researcher to also being an entrepreneur. And he is an adjunct associate professor from NTNU, leading an interdisciplinary lab within biophysics, computational and synthetic biology, whilst also being the co-founder and CEO of a startup called Syngens. So please, Rahmi, the floor is yours.
Thank you very much.
And then you can.
All right. Well, welcome all, those are here and those are online. So thank you for the invitation and the opportunity. So I will walk you through of what it means to be having a background from research and driving with the innovation activities, and more specifically, the things that we do at the company called Syngens. Syngens is a DNA design company, and our vision is to programming biology and for the products that are hopefully will meet the demands of the sustainable future. So who we are two founders, myself, as Freya introduced me, and my friend and colleague Gurvinder, who is joining online from Trondheim. He is the one who is an AI researcher, if you will, whereas I represent the biology as part of it. So our company operates in two domains: the synthetic biology, presented by me, and machine learning or AI, presented by Gurvinder. I will come into the details as to who we are, but I think I would like to put the motivation first as to why you would like to start such an entrepreneur activity, because someone from in the field, I know, and many of you are going to realize in the future, is that biotechnology will be the manufacturing technology of the future. The biology that, as we see all around us, has an extreme capacity to produce marvelous products, which we are not utilizing. And not just simply as a low-hanging fruit, but actually in terms of sustainability. The nature has taken care of that aspect already throughout the evolution, but we as humans haven't implemented that. But of course, with the benefits of the computational power and all that we are experiencing in these days, biotechnology has to be programmable, and that's the kind of the main vision motivation. The biology that we have seen today is programmable, and the programming happens through the capacity that the biology has, and the information transfer happens via the DNA. I believe you have heard DNA. Are there anyone who has had biology from before? I know that I'm talking to a high-tech audience here. Oh, not many. So quite many of you, I mean, probably all of you have heard DNA, right? So the beauty of life is so that you can communicate via DNA. So DNA contains a quite condensed information. And if you will, you may put any information you want to be executed into living cells. And this information can happen via transfer of DNA. If we have a control on how we design DNA or how we encode that information in a piece of DNA, we can communicate with life, and we can give life a certain directions to produce things that we want to produce. And that's how the programming happens. But we can't predict. We have DNA sequences, we can modify these DNA sequences, but we don't know what it will lead to, or at least we don't really good at guessing what the outcome is going to be. So specifically what we are doing is we are using biology to produce stuff. That could be enzymes, that could be the COVID vaccines that you have most of you have gotten. All can be produced by biology. So that means our product is DNA, but actually the final product is the information that is encoded in the DNA. That could be enzyme, proteins. I talk about a lot of enzymes that you're using in your laundry. Powder, for example, contains enzymes that are produced by living entities. We can use them to produce the stuff. We can isolate these things and put them into washing powders, and all of a sudden, these enzymes can eat a stain on your T-shirt by degrading it. So by all means, biology is programmable, but not predictable. So that means in terms of what we are experiencing today, despite all the technological advancements that we have, we can't design these aspects and features rationally. And I will not go into the details of the biology as to why it is so, you just have to believe me. And of course, once you don't have predictable, you can't scale up because everything goes in a low throughput fashion. Things cannot be scaled so easily. These are not the only problems, but are the most significant problems that we are targeting. Before I continue further, I would like to give a kind of glimpse, again, knowing that this is not a biology audience. If you look at a DNA, even though when you look at it, it only reads A, T's, and C's, and G's in some random combination, you don't see anything that it is controllable. Like there is no start, there is no end, it's just one continuous stretch of sequences, right? There were no white spaces, no commas, no periods, no nothing. It's just one continuous stretch of sequence. But when we look at the sequence, we can look into things like things that are doing something, which we call them coding. And there are things that are not doing anything, obviously, but actually involves in the regulation of that particular DNA. So without making the picture more complex, you can think of DNA, certain parts does coding, certain parts don't do any coding, so it is non-coding. The coding structures can be, these nucleotide means that actually there are thousands A, T's, and C's, and G's, and the non-coding part is about 100 to 1,000 in terms of length. So if you want, let's say you have an enzyme, you want to produce an enzyme, that would be the coding part, and the DNA, the part of the DNA that controls the production of this enzyme or the information, how this and when and how much this enzyme should be produced would be in the non-coding part. So play with the numbers. If you just wanted to design a DNA, which is in the length of hundreds, this would have been a total number of combination of sequence that you can play with. I mean, I think it's obvious that you can't handle this many number of single DNA constructs. It's not possible. What we do today is we are at the level of 1 or 2 constructs when we want to produce stuff, but we can't predict. So that's where we are today. We have certain regulatory elements that we know that they do work, but we can't handle the context, meaning if I put a DNA sequences together, they all of a sudden may not work, even though individually they do work or they would work because there is something called context. So that means you can't handle the complexity. And therefore, in this complexity, we are using or benefiting from AI. And that's the motivation for us have activities in this field, benefiting from the capacity of AI to read these ridiculous length of billions of thousands or certain, depending on what you're looking at, sequences that doesn't give any meaning to us. And it's also nothing something conscionable to us because we can't read these sequences like you're reading a book. And therefore, the AI provides these opportunities to us that we can read, understand the context, and based on this understanding, hopefully generate sequences that are meaningful for biology. And by designing these sequences, then we can hopefully achieve predictability and produce stuff in a predictable manner. So what we do in Syngens is, as I said now, we are practically using AI to rationally design sequences, not take things from the shelf, except that they will work. We actually design from scratch. We put these A's, C's, and T's, Z's, and G's in certain range, in certain order, and takes care of the context, means if you put things together that they will work, that will allow you to produce the stuff that you're interested. This is kind of terminology for field specific, but we can handle more other aspects in a rational manner. So I kept the part on the scientific bits more into the minimum because giving the focus of this event, how we do this transition, I would like to more focus on that one, and hopefully questions are when we talk towards the end of the day. Yeah, you may ask me some technical questions as well if you wish to do so. As in introductions you have heard, I work at NTNU Department of Biotechnology. By training, I am a molecular engineer or molecular biologist and an engineer in one level. We have to do this transition from doing an academic research to creation of a marketable product. Well, as we all know, for at least those who have an academic background, there is no one judging you in terms of what you're producing until you come to publishing. And all that run, all the grant applications, it's your mind, it's your group, nobody judges you. And that is a really hard transition to go from academic research, because that means you build stuff, years of work, 5 years of project time, so much money. and then you come to the publication stage, and there you kind of communicate with others who are judging you. Whereas when you're coming into a establishing a company, you are in day one. You have to convince people that you're delivering that has a value for your end customer. And that transition is an extremely important learning experience for us, actually, or change of minds, let's say, mindsets. Maybe not much of a challenge in terms of once you know that you have to do it, you can do it, but that's kind of has been the one obvious fact that we headache that we have seen. And me being an academician, again, I see 100,000 opportunities, project ideas to pursue in terms of academic tradition. But whereas when you're dealing with a product, you're only dealing with this thing. The goal is to make this thing as the most valuable as possible, not building 100,000 of these with different features in one go, because you need to sell at the end. Because if you have no value and you cannot generate a value after your product, there is no purpose, there is no point to it. So that, of course, that's again is a big contrast from moving from a research-oriented to or academic orientation to deliver a product that it really delivers. So those are not maybe the only points to raise, but given the time allocations, that are kind of points that I would raise. But of course, it doesn't end up there. We also have other headaches, especially in the field of biology. There is enough data, and generating data is costly and time-consuming. So one of the challenges that we do actually experience is because there isn't enough data, acquiring and curating data is pretty significant of our daily tasks, and it turns out to be a challenging. One of the beauties as well is that we are research-based. So that means if you want to generate data, we need to carry out research. Actually, that's kind of maybe going back in one way, being an academician again. But of course, you have the product in mind. You need to generate data so that you would benefit from that one. But as you see, there are some conflicts of interest as well there too. And for us, that we have, again, realizing is actually the cost-effective access to HPC is extremely crucial, and therefore maybe NORA and such organizations become valuable, not only for companies that are already rolling, but of course, those that are in the phases of initiation. You want to play with the ideas. So if we haven't had infrastructure that we can utilize with reasonable cost, we would not establish what we have right now, because having an idea in a machine learning level, it's not feasible if you don't play with it, if you don't tweak it a little bit. So that is an extremely crucial aspect too. Right, so if I have to say a few things about lesson learned from commercializing a research-driven innovation project, is that we operate in two worlds. So that means it's a dry lab. Dry lab is the things that my colleague does when he sits in front of a computer. And on the wet lab, when we get that DNA, put it in the cell, and then we see what biology thinks of that DNA. So that means we operate in two different worlds. Which is all fun and good, but the one hitting one problem that we immediately face is actually, as soon as you talk to investors, or you talk to Research Council, or you talk to Innovation Norway, specifically in Norway, then all of a sudden you're hitting a wall, because these are difficult concepts to grasp. So that means we need to be able to handle the complexity and then transfer this one to, for instance, investors. How are you going to handle that complexity? And that is, again, requires a mind change, because I am not the one who is investing. So I need to know what their worries are, what type of growth they're estimating. So that means whatever the field you're operating, you need to have different mindsets to understand, and for that matter, maybe on a level sell your idea. And this is what we have seen as a pretty significant challenge. And I cannot stress the point of talking actually about the things that you're developing, see what the perspective of others are on your product. Maybe briefly, where we have started. So we started in summer 2018, development of the platform and some algorithms. We got hold on to data on a very limited data set, just consisting of 5 microorganisms. These are bacteria. And then we established the initial platform. Theoretically, we knew that things were working, but we take the theoretical stuff in a practical biology, tested things out, and it worked. And therefore decided that, okay, there is something that we can create a value out of our ideas. And that because it was a foundation of establishing the company. That was November 2020. And from there on, and again, I cannot stress out the point of actually really early on, before we even developed any product, that you talk to final customers. You're not thinking of what you think is a viable product, but actually you're hearing what the customers do want. Issue is that sometimes customers they don't know what they want, but they have an idea of what they want. So that means this interaction is extremely important. There never should be a one level. That's another conflicting thing that I see in my life, is that as an academician, for example, you pursue your patents. You don't talk to any potential final customer. You invest 5 years, 10 years developing something, patenting, all that cost and effort. And a hard way you're finding out that actually is not viable for whatever reason. So actually there's another time, the kind of different mindset we have, that we talk really early on to final customers and hear what their needs are. And I can tell, as someone who has experienced in the field for over 15 years, I have heard things that I never thought that a company would interest it. So if we were to spend time and effort for 5 years building what I think is the product, that would have been waste. So that's the different things that we are following as Syngens. We don't have any patents. We all have is a know-how. And June, August 21, so we went from scaling up from 5 to 1,400. And of course, that required that we need to get a significant GPU hours and data storage. And I can tell actually Tromsø is pretty specialized because we have a company here, ArcticZymes. So this is our first customer. We are already delivering a service to them. So we have established, we are also in communication with different companies. And as of today, we are generating data. So that means once we have those in place, we can take the discussion further on to further other companies. And we also got, as I said, we need to generate data, and the generation of data usually happens in some sort of what you would think in a research projects. But luckily, both the Research Council of Norway and EU provide such projects where you would apply. And I couldn't stress the importance of getting hold on a soft funding if you think if like you have some product at hand, but you need to work further on. Well, with that, this is in a nutshell what Syngens is. A DNA design company, works in the field of synthetic biology and machine learning. And we design DNA sequences with the goal of generating sustainable products. And with that, I would be happy to take questions if I have time.
Time for 1 or 2 questions.
Yes. Well, thank you very much.
Could you please repeat the question? So
yes, sure.
Regarding the last thing you said about soft funding, I guess that is funding that does not ask for any ownership.
Can you repeat the last part? Not ownership?
So soft funding, that's funding that does not require ownership of the company.
No,
right?
no. So the question is, sorry.
That was the introduction.
Yes.
So my question was whether to what degree are you worried about weaponizing this technology, and also in that aspect, how large part of the company do you think of not giving out up to others?
Oh, I see. So the question is, how much of the company do you retain yourself? We're not giving out, but that is more for investors, not for soft funding.
No, basically, it's about the funding and the fear for losing control.
Right. Well, there are 2 things. So soft funding is that you don't give away any ownership, right? The state says, we need such companies and we have such a pot dedicated. So please apply. If you're eligible, you will get X number of millions, and then you run with that money as long as you last. And there is no giving away of ownership, right? So Research Council says, okay, 5 million, 2 years, go ahead. And hopefully at the end of the 5 years, you will attract customers and you move on from there. Whereas if you want to talk to the investor, the picture is different. The investor says, what is your value? What is your worth? I want to chip in with so-and-so millions. How much you're giving to me? And that conversation goes, there is no recipe. But nevertheless, you take in some investor money, X sum, and then give away X percentage of your ownership. Yeah, that's the balance that you have to play. You know, what you think in-house so many years, what your vision is and what you're going to achieve in that time frame. And then it's a kind of cards game to say, okay, I give away this much, you give away so much investment. We haven't been there yet. We are starting to approach investors right now. We play with I mean, that's what I think I can go into the difference. So we don't follow the standard route so far. So far it played well, so we are managing. So we don't do the standard of you get your patent and attract a company and move in that line. So we are working on from day one, delivering the customer. We have very limited data. We know that our system works, but we have very limited data to make a publication, for instance. From day one, we have a customer on board. That was lucky enough that we managed to do that one. We deliver direct to the customer and we're generating data through the process.
So the bonus to your customer, your partners is product.
Indeed.
Thank you for the good questions. I think we're going to continue this in the later sessions with experienced, more experienced